So good evening, everyone. Thank you for joining us here tonight for Engineered Living Materials for the Built Environment. My name is Ken Yoakum. I have the privilege to serve as the Dean of the College of Built Environments here at the University of Washington. And I'm really been looking forward to this talk. I've been hearing about the project for a number of years now and had the opportunity to preview the exhibit earlier today over in Gould Hall. And so I strongly recommend that after the talk you come over with us to Gould Hall so that you can see the exhibit as well. I am going to start by providing land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. We also acknowledge and honor the tribal nations across Washington state and the many indigenous peoples from across the country who live and work in Seattle and the surrounding regions. Tonight's program really reflects one of the core values of interdisciplinary engagement here at the University of Washington. And I'm always struck and amazed by that really some of our best and most impactful work comes when we get together, we step outside of our familiar boxes, and we begin to explore questions in new ways to generate new ideas, to generate new ways of thinking about how we can engage in this world and how we can advance advance the ways in which we know how to work with one another. The emerging field of engineered living materials, it's a rapidly evolving field, and it's combining synthetic biology and material science to mimic the properties of living materials and organisms. And while the range of the potential applications for this work is really vast, we have a great opportunity tonight to listen and to learn about one collaboration we have going on here at the University of Washington that is combining the disciplines in the fields in bioengineering, in chemistry, and in architecture. And what this team is doing is working to create novel and environmentally responsive materials that are really both resilient and regenerative in their own ways, developing properties to understand how these materials, as living materials, can thrive and adapt to changing conditions. The team's work, it's pushing technological innovations forward, but it's also expanding some of the contemporary values from within or contemporary boundaries from within our college around design, architecture, and engineering. And it's inviting us to consider what materials are, what they can do, and how they can work with us to help shape more sustainable and resilient futures for our built environments and for our cities. And this collaboration is really reflective of the potential that we can create here at UW for building the bridges across the foundational and the applied sciences for direct community impact and for advancing the public good. Before we jump in tonight, I would like to recognize a few of my colleagues who are also in the room, Deans Diane Harris and Daniel Pollack. Thank you all for being here from the Arts and Sciences. I know Dean Nancy Albritton from the College of Engineering is also a strong supporter of this work, but she couldn't be here tonight, unfortunately. It's now my pleasure to introduce our first speaker, Professor Alshakim Nelson. Dr. Nelson is the chair of the Department of Chemistry and the Job and Gertrude Tamaki Endowed Professor. He earned his PhD in organic chemistry from UCLA and completed his postdoctoral work as an NIH fellow at Caltech. Prior to joining the University of Washington, he was a research staff member at IBM, where his work focused on nanomaterials and microelectronic applications. At the University of Washington, Professor Nelson leads an interdisciplinary research group, developing stimuli-responsive materials for 3D printing with applications spanning medicine, soft robotics, sensors, and as we'll see tonight, sustainability. His work has been recognized with numerous honors, including the IBM Master Inventor, an NSF Career Award, Kavli Foundation Fellow, and the ACS PMC Young Investigator Award. Please join me in welcoming Professor Nelson.
Thank you, Dean Yoakum. It's really great to see you all here today, and thank you for coming. This is just such a unique event that we're holding because we span across multiple colleges, and in that way we have a very unique collaboration that we're super excited to share with you today. So, but this all started because we have a problem. And the problem is that there are 8 billion people in the world today, and over the next 10 years, we're going to add another billion people. And what that means is that we need to provide the living infrastructure, the buildings, the city infrastructure to support this growing population. And the problem is that our current technologies don't really support anything that's sustainable, right? So 42% of the CO2 output, global CO2 output, comes from the building infrastructures that we have, most of it from cement production. The materials that we build with, they're not recyclable, right? They don't come from renewable sources, and that's even worse in a situation where we know that the lifetime of a building is 30 to 50 years. Okay, so we're talking about a lot of waste being produced. And so now is really the time to come up with new, innovative solutions, just as what Dean Yoakum was saying. We need fresh and new ideas to start to solve these problems now, because we know they're only going to get worse into the future. So over the last 3.5 years, I've had the pleasure of working with a great team of collaborators. It spans chemistry, chemical engineering, bioengineering, and architecture. And so we've had this NSF-funded program over the last few years, where our written objective was to address key fundamental challenges associated with the integration and sustenance of engineered microorganisms into materials designed for the built environment, and to develop and train the future workforce to support these efforts. And so this evening, what we want to do is to highlight all aspects of what we propose. So we're going to give you some highlights of the research aspects, which are here highlighted in green, but we also want to introduce you to what we've done to work with students, to engage them, to think about what the future of some of these technologies might be. So before I even started working on engineered living materials, when I thought about bacteria and fungus, I would think about disease, right? And these very negative connotations associated with them. But at the same time, what I learned is that we have these very positive interactions with these organisms as well. All right, so they reside within our bodies. We depend on them for our health and digestive systems. Humans have been using microorganisms for thousands of years for food production. Right? Many of you probably make bread at home using yeast, and they're very comfortable working with these organisms. But also, we use microorganisms for large-scale production of vaccines and therapeutics today. In fact, all of the insulin that's produced in the world, comes from engineered microorganisms. So understanding that we have these very positive relationships with these organisms, how can we start to capitalize on them even further? And so that's why we've been engaging scientists like Hal Alper at UT Austin and Shota Atsumi at UC Davis for the last several years. So these are scientists who understand how to genetically modify different organisms. So our collaborator, Hal Alper, focuses on fungal organisms and has modified them genetically such that they're able to consume waste cooking oil, and transform them into more benign products. They can also use them for the degradation of commercial plastics, and also the production of different molecules like biofuels and other therapeutic compounds. Now, similarly, our collaborator, Professor Atsumi, works with photosynthetic organisms. So these are algae and cyanobacteria that can take sunlight and use that energy to transform molecules into higher value materials. So a very simple molecule that these organisms consume is carbon dioxide. Right? And so his lab focuses on using carbon dioxide as a feedstock to make biofuels or even sugars that can be used as additives in our different food sources. And so working with these very talented scientists, we asked the questions, what if we could harness these microbes for applications in the built environment? And so if that were possible, then we can imagine a world where we have smarter buildings, right? So ones where we can use these organisms to create anti-corrosive coatings. Or instead of having buildings that emit CO2, have buildings that can consume CO2. You can imagine that these buildings can undergo self-repair, right, when damage is performed, have enhanced waste treatment. You can imagine self-replication. Remodeling your home could be simpler because you could potentially grow your walls in place. So with these very big concepts and ideas in mind, what we want to do this evening then is to really take you through this path, where first I want to tell you about some of the materials that we've done at the chemistry level to create materials that we could use for 3D printing with microorganisms. And then Professor Ọlánrewájú will take you through improving our design space and understanding how we can sustain these organisms within these materials for extended periods of time. And then we want to introduce you to some concepts that Professor Proksch has been working with students here, where we present architecture students with concepts from our research to inspire them and to create these bold new ideas that, as scientists, maybe don't occur to us. And in that way, we create this incredible feedback loop between the research that happens on campus and then the training and education, and that is continuous over time. So then to begin, I just want to put up this picture of an appliance that probably many of you are familiar with. This is a laser printer, right? You find this in your homes, on your desks, in your office spaces, workspaces. But 30 years ago, some of you might remember what a desktop printer looked like. These dot matrix printers were a lot slower, they were very loud, and you could literally see the individual pixels that were being deposited. Right? But 30 years ago, we did not imagine that these desktop printers that we have today would be much faster, higher resolution, that we're printing in color over large areas, we print microelectronics. I mean, a lot of these concepts and ideas did not exist 30 years ago, but they came with these capabilities. All right, so let's imagine what a desktop printer looks like 30 years from now. Well, I would propose that we would find 3D printers as just a very typical appliance in our homes, in our workspaces, in hospitals and other areas. And so what will we be doing with 3D printing? Well, some of the ideas that have been proposed is that we'd be using them for rapid prototyping and design iteration, personalized manufacturing, especially in the healthcare spaces, and then on-demand printing, where we could create the products that you want wherever you need them, whenever you need them, even if it means doing this outside of the orbit of Earth's orbit. So tonight, I just want to focus on this idea of rapid prototyping and design iteration, because this is something that we're harnessing today, right, with these students. So we are going to provide them with the ideas of the materials and the capabilities of a 3D printer. And with that, we challenge them to use their imagination to come up with very creative ideas that initially only exist in the digital space, right? These are all virtual ideas. And with a 3D printer, you can transform them into the real world. So what we've done in my lab is to develop the chemistries to create these molecular networks that allows us to encapsulate these cells. And the way that this works is that we have these aqueous solutions where we introduce these molecular precursors. We collaborate with these other groups, such as the Alper lab and the Atsumi lab, to incorporate their engineered organisms into our resins. And we use a light-based 3D printer to be able to create a three-dimensional construct. And within this construct, what we've done at the nanoscale and the microscale is we've made this molecular net. Right? It's this three-dimensional net that traps the cells so that we can keep them where we want them to be within this printed structure. But at the same time, the cells are viable and they're metabolically active, they're alive. And so this is all done within a water-swollen network, so we typically refer to these as hydrogels. So key points here is that we use the 3D printing that controls the shape, so we can create whatever design we want as long as we can create a virtual design using CAD. And then with these engineered living materials, where we've printed these structures that encapsulates these organisms, what we have are these materials where their function comes from what the organisms can do. Right? And so we have to preserve the metabolic activity and the functions of these organisms in order for these materials to work. So just to highlight then, the challenges that we've been working on are first, can we create hydrogel structures that have mechanical stability? Right, so when I say hydrogel, classic hydrogel is Jello. Right, Jello is very jiggly, but you can't really make things with Jello. It's too soft. So we need that type of structure, this water-swollen structure that we can build things with. We also need cell viability. So without the cells being alive, these materials won't have function. And then we also need to preserve metabolic activity of the cells. So they need to be able to produce the different proteins and enzymes for them to perform. So what I want to do over the next couple of slides is just highlight how we've been able to do that to address these challenges. So I don't want to spend too much time talking about the details of how we did this. That's like a whole nother hour lecture. But this is how our light-based printer works. So we're projecting light through the bottom onto this liquid resin. And this is an object that we print layer by layer. So for each layer, we're using light to draw a pattern. And you can see how fast that laser is rastering around. So when we think about a new material or new resin that we need to develop, the two things that we focus on are the viscosity of that resin, but then also the rate at which we can create these networks. All right? So with those brief light exposures, we have to create that molecular network to trap the cells. Now, our approach has been to use protein-based resins. And so the advantages of using globular proteins for 3D printing is that we would be using and developing materials that are biocompatible, sustainable, and biodegradable at the end of its lifetime. Okay, so details about this. So we're trained as professors in this room to know that that bell happens, right? Yeah. All right, so we can print these protein-based materials. And so here what I'm showing is side-by-side comparisons of our materials, these water-swollen protein-based hydrogels in orange, and then the commercial structures that are clear right next to them. And so when my student Patrick Smith first produced these hydrogels, he said, yeah, these are incredibly stiff. They have a compressive modulus of 6 megapascals. So what does that mean? And so I said, Patrick, put some weights on it so we can understand what that means. So he went to the gym and he put nominally 20 pounds of weight on this hydrogel, but what's important is that there's no sign of mechanical damage, right? So this is a water-swollen structure that can support that sort of weight. This is not something that you can do with Jello. But beyond that, we can now start to use the 3D printing capabilities to now produce these water-swollen hydrogels that are mechanically functional. So we can create this nut and bolt and start to assemble these things together. Right? So what you cannot see at this scale is that there are cells that are trapped within these hydrogels. Right? So these are actually living materials that we are now creating into these living machines. So they're mechanically stable. The cells are viable, and so we can perform these scanning electron microscope experiments to show that at different regions of these structures, we do see proliferation. So not only are the cells alive, but they can also grow if they have space. And then finally, these cells are metabolically active. And so what that means is that for a printed structure, we provide it the nutrients, the cells, the nutrients that they need. They transform those nutrients into other products. And so as one example, I have shown here along the x-axis is day, so that's time, and we're looking at the production of this molecule called L-DOPA over time. And so we see that those bars are increasing over this time period, so the cells are continually creating this L-DOPA within these materials. At the same time, it happens that with L-DOPA, these materials become stronger, and also it turns black. Okay, so we know that these are metabolically active. So what we've been able to do then is, depending on the organism that we introduce, we can create engineered living materials that become stronger over time. Right? So the longer that we feed these organisms nutrients, the stronger the material can become. We've also created elms that can biodegrade on demand. So this is a protein-based network. We can provide cues for the organisms to produce enzymes to degrade these materials. We can create elms that can consume used cooking oil, which is something that we will see later on this year in the published literature. And then finally, we've developed elms that can consume CO2 and produce biofuels. And so these are just some of the baseline capabilities that we've shown over the last several years. And so just to summarize what I've discussed so far before I hand it over to Professor Ọlánrewájú, we have 3D printed elms using protein-based networks. They're mechanically robust, the cells can be preserved, they're alive, and they are metabolically active. And so the next big question for us is, this is great on a lab scale, right? But we're talking about deploying this out there in the environment. So there are several challenges to that, some of which include, how do we sustain these cells for extended periods of time, beyond just a few days? And then, what will it take to deploy these types of structures in the real-world environment? So with that, let me hand it over to my colleague, who will tell you some of the concepts that we've been working on.
Thank you so much for that wonderful introduction, Professor Nelson. It's good to see you all. It's such a joy to do this. It's very much a unique experience, especially now. And I'm excited to take the baton and tell you about the next step. So how do we sustain these elms in the real world? So let's do a thought experiment first. What if you had to live in your own food, water, and waste? Like, what if that's how you lived all the time? It's gross, not just in the Matrix. It would be gross in this world too, right? And that's what we currently do to elms. This is how most people make their engineered living materials. It works well. You can do certain things with it. You can make biofuels with it. You can do a lot of really important things. But the reality is they still live in their own soup, right? And so where I came in was 4 years ago when I started my lab at the University of Washington. I was but a lowly plumber. And Professor Nelson was like, hey, how would you like to make plumbing for microbes? And I said, my day job is I make medical tests. And so this is a video of one of the tests that we make. This was in the COVID era where the only thing you could do was make COVID tests. And we're trying to see if we could make them better by adding more plumbing to them so that you could have more liquid handling steps. And when we do our job well, most of the time you just want to forget about us, right? You don't want to be thinking about the plumbing in your house. You don't want to think when you flush, where's that liquid going, right? And like, will it make it there? We live in Seattle, so a lot of us have experienced leaks. We had a leak not too long ago. When things don't work with the plumbing, you notice, right? So I said, well, let's do this. I don't really know what you want me to do, but let's try. And a lot of my inspiration as a plumber came from watching Inspector Gadget. And the lesson I learned here, if you've never watched it, is you use the tools that fit the problem at hand, right? And so for us, it meant, you want me to make plumbing for bacteria. Well, let me use tools that fit this problem at hand, right? So what's the relevant size scale? Right, so our microbes are somewhere in here, right? Like about a micron, depending if you're looking at yeast or if you're looking at E. coli. So how do we fit plumbing for this? How do we make microplumbing? So I'm going to give you another scenario, right, where really what we're trying to do is move liquids around or manipulate liquids in unusual ways. And one scenario is this. This is a water strider floating, moving around, living its best life, zipping along. Another scenario, this is Chris Hadfield on the ISS, International Space Station, squeezing the towel, and instead of the water coming off, the water just sticks, right? And this is in between playing songs on the ISS, because it's a good life. And so my question to all of you is, what do these two scenarios, where we're making liquid do unusual things, what do they have in common? I think I hear it. Surface tension. It's that they use the force. Capillary force, surface tension, right? This is the idea that the cohesive forces between liquids can do very unusual things. And this becomes pronounced as you move to smaller and smaller size scales, right? As these microchannels, or as these channels or tubes get smaller, capillary forces become dominant, and they kind of start to defy gravity, which is part of what you're seeing in space, right? When you're in space, gravity plays a much smaller role, and all of a sudden squeezing the towel becomes a big issue. Washing your hair becomes a bigger issue. And so one of the things we thought about is these microbes are small. How do we use the force to support plumbing here? How do we set it up so that you don't have to worry about plumbing, but the structures and the 3D geometries that you build can essentially plumb themselves, so you don't even have to think about them? So Professor Nelson nicely set this up. We used a 3D printer to make these small and complex geometries that we need. And some questions I want to ask all of you, just as a quick show of hands. Who in here owns a 3D printer? Okay, like, say maybe 10% of the room. Who in here wants a 3D printer? You get a printer. No, I'm kidding. I don't. Lean times for academia right now. I wish. However, most of you, when you think of printers, probably have one of these, right? You're thinking something like this. It's a filament-based system. And here's how they work, right? Basically, you're like extruding something with heat into filament, and you're basically making something. You're literally printing something out, is the image that most of you have in mind. And one key thing I want to point out is that not all printers are created equal, especially when you want to do tiny plumbing, right? If you want to get to the size scales where capillarity is dominant, you can't just use any printer. And I'll tell you this because not only me, but several people have tried in the field of microfluidics, which is the field I come from. And the idea is you start to see things where if you use your filament printer, you see these gaps, and if the role of these conduits is to hold liquid, you basically get leaks everywhere, right? This is an inkjet printing system that's very similar. Some of these printers are hundreds of thousands of dollars, and you would think they would work really well, and they're good for some things, but at some of these size scales, they don't really work that well. The one on the right here is a resin printer, and we'll talk about what that is in a second. And as you can see from these images, if you want this thing to hold water, you maybe want to use this one, right? So use the tools that fit what you're trying to do. And this is an illustration. This is from a group in Tasmania. They actually recently had the biggest microfluidics conference in the world, went to Australia. There were a bunch of people in Tasmania with koalas and kangaroos, and it was very cool. But you can see that if you just want liquids to flow in very predictable ways, if you're using your filament printer, you get all these artifacts that you don't want in there. So how do we do this resin printing? It's akin to old school photography. If anybody has done clean room type work or like dark room type work, the lighting has to be controlled because really light is what changes your color or shape. Light is what writes for you. And some of the printers that we use, you'll actually see this at the exhibition hall. This yellow cover here is precisely to control the wavelengths of light that get in there so that things don't cure and take shape until you want them to. And this is just a schematic of what that looks like. It's basically, as Dr. Nelson said, you have a laser, or sometimes now, even just a piece of an LCD screen. You put it under your photosensitive material, and then you shine light, and you do that over and over again. And so it's a layer-by-layer process. You go back down, you turn on the light, goes back down, turn on the light. And basically, you can do this over and over. It takes us in our lab maybe about 15, 30 minutes to make a prototype. And these printers are getting more and more inexpensive. I think now we can buy a printer that can do something 1 or 2 times the width of your hair for $200. So the prices really crashed, a bunch of patents expired, and there's all kinds of companies. Every time I go to the website, there's a new sale. There's like a Father's Day sale, Easter sale. So in my lab, actually, I'm telling my students, you get a printer, and you get a printer. And this is our current workflow, right? So we do a design, because we need to make sure we're making something deterministic, that we understand the fundamental principles, the size scales, the shapes, and you can do very weird things if you can control the force. So we do our design, and then you slice again to get these layers. And once you've sliced, then you have to think about the resin formulation, which is where it's excellent to have wonderful collaborators in chemistry that can pass you things that they've made. So we don't have to reinvent the wheel. Then after that, these materials sometimes contain protein. They have other things in them that are essential. Then we do the 3D printing. And then we characterize them under a microscope, because these things are really small. And at the scale that we're working on, we need to look at them, use the tools that fit what you're trying to do. This is some of the sample characterization that my student Aileen often does. We do this almost every day now. Well, maybe not every day. Aileen would say this is a lot of work. I don't want to do it every day. But what this graph is showing you is what we expect the size to be and what the size was measured to be. And this is showing us that we can print things about the width of your hair fairly reliably, reproducibly, with these engineered living materials that can sustain microbes. So this tells us we're in the size scale where we can start to make things flow just by themselves. And so we want to show that. This is a video where Aileen is adding dye that's added to bacterial growth medium. And what you're seeing is spontaneous flow without us touching it, just based on the size of the channels, showing that we can guide fluid into these microchannels. And then I'll skip ahead and just show you that part of what we're also trying to demonstrate is perhaps you can have a reservoir of fluid that then guides liquid to particular regions, and then you can bring in nutrients that diffuse all through the materials and keep them happy. And then you can replenish it at dedicated periods of time. So instead of living in your own soup, you can have a mouth. And then you can put liquid in your mouth, and then it goes where you want it to go, right? And then if you look at this after a 24-hour period, you can see that it's really gone through and provided some sustenance, presumably. In this case, we're just looking at dye. So we're demonstrating pump-free autonomous flow of dye-incorporated growth medium, really as a proof of concept that maybe we might be able to do something that resembles long-term self-sustained hydration in elms. The other thing that we've been doing recently is we can take some of the developments that have happened in the world of microplumbing, separate from engineered living materials, and say, can we bring that into engineered living materials? So this was a paper that made a splash in my field. It took inspiration from trees, how transpiration occurs, how there's evaporation up top, and then how you can get this spontaneous flow. And we saw this and we said, well, can we do this with engineered living materials? Of course, these protein materials are a bit like Jell-O. The material properties are different. They kind of swell, things go all over the place, but can we do this? So we tried. The beauty of 3D printing is you can find out very quickly. A lot of the time it won't work, but when it works, it's really beautiful. So this is us adding, again, dye-incorporated growth medium to lattices, and it works, spontaneously wicks. And we're starting from smaller scales, but these unit structures, the beauty of them is that you can start to make arrays of them and make them relatively large. And if the unit cells are small, then you can really get that spontaneous flow. One of the things you'll see at the exhibit is that some of the architecture students are thinking about how can we make these resemble materials that actually would work in places that people would be. And I'll let Professor Proksch tell you more about that. I also added a blooper because working with microplumbing is not that simple, right? Actually handling these things, every now and then they fall over, and then you try and get them back up. They're kind of sticky. It's a lot of work. So I think one of the things we're also training our students to do is to do hard work, to have good hands, right? So just like real plumbers, I'm a real plumber, just like macro-scale plumbers need good hands, micro-scale plumbers need good hands too. And if you can work at this scale, then you can do almost anything, right? So I think some of the skills in baking and plumbing on these things are fairly transferable, and it builds a lot of resilience to failure. So what are we doing now? Students in my lab and students in Professor Nelson's lab are saying, can we actually grow things in this? Because so far I've just shown you food dye, and I love that you all believe me. But as scientists, we have to then say, do these things, I hope you believe me, do these things actually grow the yeast that we want them to grow? And so we're putting them in little culture dishes, we're adding a bit of medium at a time, putting them in incubators at the right temperature, and then we're watching them grow. And then we're putting them under the microscope and looking at cross-sections of where those engineered microbes actually show up. This is work in progress, but we're very excited about what's taking place, and we have plans for how we will share this with the world in the scientific literature and also in opportunities like this, presentations like this. I'm going to summarize and then hand over to Professor Proksch. So I hope that I've convinced you of the importance of plumbing on a macro scale and a micro scale. I hope you believe that we can get spontaneous flow of liquids into these systems. And I think the question then is, if we can start to think about deploying these systems, what do we do with them? I think one of the things that we all talked about before this presentation was, what do we want to leave you with? And it's this idea that we want you to understand the length scales and the time scales of this work, going from molecular to more micro scale, and then to macro scale, and going from what we do today, tomorrow, in 5 years, in 10 years, maybe 100 or 500 years. So I'm going to leave it to the architect to give you that big picture, and I'll hand it over to Professor Proksch.
Thank you, and thank you everybody for coming. It's exciting to see such an interdisciplinary audience here. So what's the architecture perspective? How do we now integrate these elms into the built environment? To contextualize this a little bit for you, for those that are not in architecture, architects are fascinated by working with living systems. And they've done this at various systems, and this one is one that's maybe close to what elms do, because they work also with microorganisms. These are photobioreactor facades where algae is basically producing biomass, but it's not a material. We are building an infrastructure around the cells that live in, they are the free cells that live in nutrient solution, and they will pump through this. So very different, fascinating but very different. Another field that has taken a lot of traction in architecture recently are biogenic materials, materials that are produced by living cells, and mycelium-based materials, bacterial cellulose materials, maybe not yet as far for the application, but bio-CMUs, where the cells generate the cement equivalent to hold the aggregate together. So some of these are actually on the market. What is very different from our materials is that the cells are not active when we use them. They're usually dry materials. They behave pretty much like our conventional materials, and people are fascinated by it because it is much more environmental, but they are very different than our living. Not all of them are living, but the ones that we will show you in the exhibition have all living cells. So my statement here, polymer-based elms are new frontier for design integration, because we have to think about how to keep the cells alive. We have to hydrate our materials, which is sort of counter to what we learn, keep a building dry. So we have to feed them, we have to provide gas exchange, and so on. We have to, for the interdisciplinary collaboration, we have to transfer knowledge between scientists to architects or built environment experts, think at a cellular scale. But we get these specialized functionalities, and we learned a little bit about a few of them, and there are more to come, so it's very exciting, and architects bring a lot of the core skills that you need. You need to know how to do computational design, you need to know how to 3D print, digital fabrication, and have built environment expertise. So that thinking led us to, when we started working together, to say a research seminar and studio in architecture, which is basically the equivalent to a thesis, would be a great place to have students work with this and be a laboratory where they can experiment with the design implementation. And now 3 years into this, we thought, we learned a lot. and we would like to share that with a much larger audience. And that's why we started to conceive and create this exhibition, which has had a jump start at the end of last spring quarter, and now we have a formal exhibition, and we're really excited about this. So just to give you a little bit more detail about this, where the two the research design overlaps. So if you see this very abstracted iterative process that the research teams go through, the architecture student can plug in, or architects in general can plug in when there is the idea of prototyping. That's what they are equipped to work with, 3D print ideas. And of course, on the left side, the whole idea about how to integrate this in design. So in two places of this iterative process, architects can be really part of this investigation. And I said, we are in year 3, so we have a little bit of experience, and I will share now 4 high-level findings that we think make a big difference in developing a form of knowledge sharing pedagogy. How can non-scientists be active partners in this investigation? And where are the 507 students? Raise your arms. We have an active course. Don't be shy. Yay, you're all here. They are working on this right now. So it's all a little bit still like, oh, what should we do? But they're making great steps forward. So the 4 steps, or 4 observations. We started now with a selection of the microorganism. The chemistry labs work with several organisms. Some were introduced to you, and we selected 2, the Yarovia, the oil-consuming, waste oil-consuming species, and then cyanobacteria. And all the work that you will, or almost all the work that you will see in the exhibition uses either of the 2. The second one is we need this knowledge about the organism to be easy understandable for non-scientists. And that led to really successful peer-to-peer learning when the PhD student became our TAs, and they started to discuss this with architecture graduate students. The architecture graduate students want to talk way more with the TA about the material and the microorganism than with me. And so I think that's a great success. And these 2 versions here were actually created by PhD students for the architecture students, so that we can have a kickstart with all the necessary knowledge in a place. And a great observation is that chemists like visualization as architects. I'm so impressed every time I see their publication, I see their presentation. This is so refreshing to see that they work with pretty much the same means of visually communicate. So that was great for all of us to discover. Then the computational design. The students don't necessarily have fabrication skills in computational design, and therefore a little bit of a different introduction to computational design was necessary. Here are some of the model material samples that the students produced. And then the 3D printing, you learned a lot already about this. The one thing that I find the most interesting, I see we have the same photograph, that printer you see in the exhibition. We're using the same off-the-shelf printers. Architects and all the chemistry labs print with the same relatively inexpensive printers that for the architects, of course, do a great job, but you mentioned that they even, for your precision printing, work really well. And on the right side, you see what the students do. You will find in the exhibition the active table, where we put all the small study models. These are things that we collected over the last 3 years, but a large number is already from the active course, so we will fill the table. By the end of the show, the table will be full of study models. So it's an ongoing process. And now, to give you a little bit more introduction to what you will see, but also to show how we could actually integrate these engineered living materials into the built environment, I will show 5 student projects. These student projects are all from last winter and spring quarter. So let's get started. So as I said, 2 organism Yarovia project from Niklas Espin. And he develops an entire space that has a bioheater. The project is called Bioheater because when the microorganism consume the waste oil, they produce heat. So you could use that, and you see how the computational design supports that. You could use that to heat an environment. We asked the students to think at different scales. Many of the work you see this year is at the relatively large scale, but we also asked for like a small handheld object and smaller application, and then this larger infrastructure. The second one, Yarovia project that I want to introduce is one that uses wastewater or uses the engineered living material to clean wastewater in the urban context. And these are the harbor steps where this installation climbs up the hill. And here you see the functionality of this one. There are different strains of microorganisms needed to absorb different pollution in wastewater. The infrastructure or the different elm printing types account for different collection of these pollutants in wastewater. And then the second set of projects use cyanobacteria, mainly, if I describe this in architecture terms, as facade systems, but you will see they are much more inspired than just facade systems, like this one by Christina May. And here you see well how the different computational structures are used to build a wall panel. The outside of them, of the panel, indicated in green, is a photosynthetic active elm, and the structure inside is a elm that is self-strengthening. So over time, these facade panels or shading, because they cannot be an enclosure, elms cannot form enclosures, but they can shade, they can be a rain screen. And so you see how something like that, a very lightweight structure, could be constructed. And then the second one is taking the same idea into a much more infrastructural scale to propose green roofs that replace the soil or substrate-based green roofs with elms. And Julia Cedo has biomimicry ideas to then build a layered structure that is in a cage so that you can walk on it, at least for maintenance purposes. The green part, again, is cyanobacteria-infused elm, and the substructure to it is the water collection system, so that in a green roof situation where you have intense sun and heat, the living cells can survive. And it can be applied to different roofs, of course. And then the fifth one, the final one, is Rachel Tyler's cyanopavilion called the Urban Arboretum, because it generates the same benefits, environmental benefits as a tree, but can take various different shapes. It's a self-supporting structure where the cyanobacteria infused elm is covering a structural member so that it can be a self-standing pavilion. These and others you will see in the show. And to give you a little bit of an introduction to the show itself, it's organized in 3 spaces. The science space is where you find all the great work the chemists and engineers do, and where you learn about these different steps that are necessary in our shared iterative design process. So you learn about the living cells, computational design strategies, the printing table, and you will see their publications. And then the student work is the Yarovia space, where you see the project using that organism, and then in the third space, you will see all the cyanobacteria. And we are very grateful about an exciting team. This is the active team in spring 2025, so of course it changes. So these are the all the members that actively worked on creating the first set of material for the exhibition, and each of them provided one photograph of their work with elms. So that's how this came together. And then finally, we would like to thank all people that supported this work, that worked with us at various points. And thank you to all of you for coming. But before we close it, we also open up now for questions.